So a little bit of an outline. I want to firstly introduce sensory neuroscience, and then move on to something called sensory acuity and the 2-point discrimination test. So I think you've been sent a Word document kind of describing what this is, and you might have had a bit of time to test this out. If you haven't, that's absolutely fine. You're welcome to test it out after the talk if you find it interesting. And then move on to something called the sensory homunculus. And then lastly, look at neuronal plasticity. So that refers to how the brain can change when after injury or something, or if you overuse one of your fingers. So yeah, going into too much detail, but yeah, so how the brain changes. You might have seen, so this, I've added a few questions that we can do throughout. So if you're able to either scan the QR code, or if you just type in Vivox into Google, you can put this ID code in, and then it should take you to a screen that where you can see the Vivox for this talk. And then throughout, I'll open the polls, and then we'll be able to do the questions. So I can wait a few minutes or wait 1 moment to wait until you've got onto the Vivox session. So it should be open. Okay, so sensory neuroscience, I just want to introduce this topic firstly. So what this means is how the brain receives and processes information from all the different senses. So this is really important because we need to be able to kind of pick up things in the environment and somehow behave in some way. So for example, if you were to taste something really sour, you might then in future want to avoid that. Or if you hear a really loud noise, you might want to cover your ears or move away from that sound. So we need to be able to pick up all of these signals, somehow represent that in the brain, and then behave in some way to respond to it. So as I'm sure you'll know, we've got lots of different senses. So there's touch, which in neuroscience, we refer to something called somatosensation, smell called olfaction, vision, hearing, which is called audition, and taste called gustation. So all of this different information is represented in different regions of the brain. So if we just look down here, we have different areas of the cortex. So the cortex is just basically a word that refers to the outer layer of the brain, and there's different regions of the cortex. So you have the somatosensory cortex. This is to process touch information. We have the gustatory cortex to process taste information, visual cortex for vision, auditory cortex for hearing, and olfactory cortex to process information based on smell. So this talk will focus just on the somatosensory cortex, and this enables our body to sense external changes in the environment and allow us to respond in an appropriate way. So why is this important? So we need to be able to effectively move around, so effective locomotion. We need to be able to detect the terrain that we're walking on, or kind of feel what's around us in order to effectively move. We can't be bumping into things all the time. We also need to be able to identify objects and surfaces in order to safely navigate an environment, and also avoid pain. We don't want to be touching things that are really sharp, that will cause us damage in some way. So this is a really important sensory system. So I'm just going to go into a little bit of detail about how touch information is processed and how it goes from your hand or from your skin all the way up to the brain. So this is going to be a little bit technical, but I'll try to go through it as slowly and clearly as possible. So what happens is you have something called a sensory neuron. So a neuron is basically just a cell of our nervous system, and it innervates all different parts of our body. So imagine here we have our hand, and it's innervated by this neuron, and at the end of it, we have something called a receptor. So a receptor basically picks up some kind of signal, and when it does that, it transmits that information all the way to the brain. So we've got this receptor in the hand, and in the context of touch, they're called mechanoreceptors. So they respond to mechanical stimuli. So that means if something was to touch your hand, it's going to stretch or deform the skin slightly, and that's going to open these receptors. So sensation of touch starts by stimulation stimulation of receptors found in the skin, and stretching of the skin opens these mechanically gated channels. So if you're familiar with kind of cell biology, you'll know that cells have this membrane, this phospholipid bilayer. And so this is the outside of the neuron or the cell, and this is the inside. And what happens is when you open this channel, so when you stretch the skin and the channel opens, you allow positive charge inside the cell. So when it's just kind of resting, there's more positive charge outside than there is inside. So when you open this, it's going to diffuse, it's going to go down its concentration gradient into the cell. And when that happens, it kind of charges up the cell. So it makes it become something called electrically excitable. And when that happens, it fires an action potential. So the signal is transduced, so that means just changed from a mechanical signal into an electrical signal. And you can imagine these action potentials like the language of a neuron. So when it fires an action potential, when it is kind of excited, it's kind of like it's then talking to the brain and saying something's touching this part of the skin. So it's just kind of how the neurons are communicating. And so when that happens in this receptor here, that's going to transmit the signal along the sensory neuron, and then it will reach the brain. So this is a touch signal, so it will be kind of represented in the somatosensory cortex, as we said. So the signal then reaches the brain for processing. So we've kind of discussed how this happens with touch, but there are all different kinds of signals for the different senses. So we've said that it's a mechanical signal for touch, and it's also a mechanical signal for hearing. So what happens is you can imagine sound as a wave. Sound is a type of wave, and the air particles are kind of acting as a mechanical signal, and that leads to transduction from this mechanical signal into the electrical signal to then get to the brain. We also have chemical signals. So in terms of taste, you've got these things, so taste buds on your tongue. What they do is they pick up molecules of food and then transmit that to the brain. So if you say had a sugar molecule, if you were to eat something sugary, a sugar molecule would bind to a taste bud on your tongue and transmit that as an electrical signal to that corresponding part of the cortex. Same thing for olfaction, for smell. So if you had a volatile, so that just means it's in the air, a volatile molecule. If you're smelling something, say you're smelling something really sour, that sour molecule will go into your nose, bind to a receptor, and then again transmit that signal to the olfactory cortex responsible for processing smell. And then lastly, for vision, that's a different type of signal. So that's taking in light. So it's a type of electromagnetic signal. And what happens is it's transmitting that light signal into an electrical signal again. So there are these different types of receptors to do that specific thing. So we've discussed mechanoreceptors, also hair cells, taste buds, olfactory receptors, and photoreceptors. So these are picking up all the different signals that are outside in the environment and then representing that in the brain. So these mechanoreceptors that we've discussed for touch, there are actually 4 types of these found in the skin. And they all pick up different types of information that we need in order to kind of identify what an object is. So we have mechanoreceptors called Merkel discs. So these are found very high in the epidermis, so very kind of close to the top of your skin, and they are used for the perception of shape and texture. We have Meissner's corpuscles. They're found a little bit lower, kind of in the upper dermis, and therefore motion detection and grip control. We then have Ruffini endings, so they're much lower in the dermis for skin stretch and pressure. And lastly, Pacinian corpuscles, which are found even deeper in the subcutaneous layer, and they're detecting things like vibrations. So I don't want you to kind of memorize all of these things. I just want to kind of portray that we've got these different types of receptors that are found in the skin, and they're picking up different types of information about an object. So you need to be able to know whether it's what kind of shape it is, whether it's moving, whether it's still, and its size. So all these different types of information. So I want to go into this in a little bit more detail now. So how do these different features of objects, how are they represented by these receptors? So how does this receptor know if an object is still or whether it's moving. And they achieve this through something called adaptation. So if we just look down here, if you can see, we have a neural spike train in the stimulus. So this representation is a little bit confusing, but if I just talk you through it. So these lines here, they just represent an action potential. So when you've got a line here, it's like the neuron is active and it's sending to the brain that something is on the skin. So with this first one here, it's firing lots at the start and then it's silent, so the neuron is not active at all. But then here, the neuron is active for the whole time that the stimulus is present. So if we just look down here at the stimulus, imagine this is, say, a marble that's rolling on your skin and then is staying still. So down here, the marble is rolling because we have an increase in the stimulus, it's moving, and then the marble is just still as it goes here. So hopefully that makes a little bit more sense before I move on to a bit more information. So these skin receptors exhibit two types of something called adaptation in order to achieve this. So adaptation just means it's able to respond kind of quickly, it's able to change its response. So we have receptors that are rapidly adapting and slowly adapting. And these rapidly adapting receptors sense movement, and the slowly adapting ones sense when something is just constantly on the skin, when it's just still. So the rapidly adapting receptors, these are the corpuscles. So these are the Meissner's corpuscles and the Pacinian corpuscles. So how they do this is when an object is moving, so as I said, this kind of slant here, that's showing that the stimulus is moving. And when that happens, you can see this neuron is active, so it's firing lots of action potentials. And the same here with the Pacinian corpuscle, it's active when the stimulus is moving. But then when it's still, it's completely silent, so it's not active. So it's not really signaling, it's not telling the brain anything that it's still, but it is when it's moving. So it's good for motion detection. But we also need to tell the brain that something is just sat on our skin and that it's not moving at all. So to do this, we have these slowly adapting receptors, and these are the other two. So these are the Merkel cells and the Ruffini endings. So these receptors continue to fire. So what that means is they fire these action potentials, they're electrically excitable for the whole time that the stimulus is present. So the combination of these different adaptation types allows the skin to respond to a variety of stimuli, enabling accurate sensory perception of the environment. So that just means we can detect if something is moving or if something is just still on the skin, because we've got these different types of the receptors that are saying to the brain, this thing is moving, this thing is still. Okay, so now I want to move on to something called sensory acuity. So this is a very related topic to what we've been talking about, but it refers to how precisely two separate stimuli can be distinguished. So, and this sensory acuity varies across different parts of the body, and it relates to something called the size of the receptive field. So the receptive field refers to the region of the body that a particular neuron responds to. So if we just look at the diagram below, this neuron here, as I showed in that diagram where I explained the transduction, we have a neuron that is innervating, so it kind of goes into a specific part of the skin. So when you were to touch this region of the skin, this neuron would then become active and then would signal to the brain that this part of the skin is being touched. And the receptive field is the area of the skin that this neuron responds to. So anywhere in this receptive field that you touch, if you were to touch here, if you were to touch here, this neuron would become active. And so this would have kind of a low sensory acuity because it's not able to distinguish whether you're touching here in its receptive field or whether you're touching here, because regardless, it would just become active. But if we just look here on the right, we have two neurons and the area of skin that they innervate is much smaller. So if you were to now touch this part of the skin and then this part of the skin, the brain would be able to distinguish between those 2 points because you're touching in the receptive field 1 and you're touching in the receptive field 2. So smaller receptor fields provide more precise information and contribute to a higher sensory acuity. And then conversely, larger receptor fields contribute less precise information. So now this moves on to the 2-point discrimination test that you might have had a quick read on before. So what this does, it's a kind of measure of sensory acuity, and it shows how finely the skin can distinguish between two separate stimuli. So what you need to do this is you need the ends of a hairpin. So if you don't mind ruining a hairpin and kind of opening it up a little bit so it's kind of got two separate points, or you can use two toothpicks or your fingers or something, or a compass, something that's got kind of two fairly sharp edges. And if you have someone to do it with you, a parent or a friend or something, if you lightly touch 2 points to the skin at the same time and ask the person whether they feel 1 or 2 points. So the person that you're doing it to is, if they don't look while you're doing it, then if they guess whether it's 1 or 2 points, if you adjust the distance between these two points. So test this out on different parts of the skin. So you can test this out on the back or the hands, and then adjust the distance between these points. So what you want to do, your results of this are to observe how the distance needed to distinguish between these points change. So for example, if you've got about 2 centimeters difference or 1 centimeter difference between these 2 points, are you able to say, oh, that's 2 points touching my hand? And if you had the same size on your back, would you be able to do the same? Would you be able to say, oh, that's 2 points, or just detect that it's 1? So this is asking which areas of the body have a high sensory acuity. And so I've said this ability changes all over your body, and it relates to the size of the receptive field. So as we've said here, if you put these 2 points within the same receptive field, that neuron wouldn't be able to distinguish between the 2 points because the neuron's just is either active or it's not. But for example, if you had it in 2 different receptive fields, you would be able to perceive that as 2 points because they're 2 different neurons are then activated. So I'll just do the first Vivox quiz if I just open the question. So, which part of the body do you think will have the highest sensory acuity? No worries, if you get it wrong, you can see. So I've got 80% fingers so far and 20% face. Wait a few more moments. So we've got 75% fingers, 25% face. Okay, I think that's probably also, we've got 71.43% fingers and 28.57% face. Okay, so that is very, very good. So both the fingers and the face do have a very high sensory acuity. And those who put fingers, that is correct. So the fingers actually have the highest sensory acuity, but kind of close second is the face. So you're all right, really. Okay, so yeah, so this is just a diagram showing how the different thresholds change throughout the body. So the threshold just means at what distance can you distinguish between 1 point or 2 points. So yeah, the fingers have the lowest threshold, which relates to the highest sensory acuity. And then, yeah, so the areas of the face, that's also very, very high sensory acuity. And then areas like the back and the calf and the legs, they've got a very low sensory acuity, but a high threshold because you need a large distance to distinguish. And sensory acuity also changes with age. So the threshold increases as you age. So you actually get less able to distinguish between 2 different points as you get older, which is quite interesting. Okay, so we've now looked at how touch is represented by the skin. So how it's represented by these mechanoreceptors, but then what happens when it reaches the brain? So as we've said, the somatosensory cortex is the part of the brain that processes this information. And what happens is the brain maps our body into this area. So The next image I'm going to show you is something called this sensory homunculus. So it looks a little bit strange. So this is Latin for little man. What this is a visual representation of how the body is represented in the brain. So obviously it doesn't look like a normal proportional body. We've got massive hands and we've got a massive face. So this is basically a visual representation of how the body is mapped within the brain, and it was created someone called Wilder Penfield in the 1930s. So it's depicted by a distorted human figure with body parts disproportionately represented to reflect the amounts of cortex dedicated to process that information. So because we need a lot more information about the hands and the face, there's therefore more brain space to process that. So it's just representing that the hands and the face, yeah, have much more brain power. Okay, so another VBOX now, true or false? Receptor fields in the back are smaller than in the fingers. So we've got 100% false so far. Still 100% false. Yeah, so that is absolutely correct. So receptive fields are not smaller in the back than in the face. So as we've said, we've got higher sensory acuity in the fingers, so we have really small receptive fields in order to distinguish different points. Okay, so the reason why we have more brain space is because we've got a greater density of these receptors, and they've also got smaller receptive fields. So this is, yeah, just kind of reiterating that point. And then smaller body parts on this homunculus, they have fewer receptors in that part of the skin and then less brain space represented. So why is this? It kind of is intuitive, it makes sense. We explore our world with our hands, so we need to be able to pick up as much information from our hands as possible. It's not like you go around kind of exploring the world with your back or your leg or something, so you need a lot of brain space to process that information. And the same with your face. The face is obviously very important, it's protecting your brain, so you need to be able to detect and process if things are touching the face. So I said that this guy called Wilder Penfield, he discovered this homunculus. So I just wanted to go into a little bit of detail about how he actually did this. So what he did is he performed electrical stimulation of the somatosensory cortex. So what he did was, there are these tiny little things called electrodes that can be put into the brain. So they don't really damage the brain at all. They're very, very tiny. And so I've said that neurons are electrically excitable. They use kind of a form of electricity in order to communicate. And if you put a small amount of electricity into the brain, it's kind of mimicking that part of the body is being touched. So if I touch my shoulder, the shoulder part of my homunculus would be activated, and it's kind of the opposite way around. If I put some current or I put some electricity into that part of the brain, I would kind of have a sensation that someone's touching my shoulder. So this will cause somatic sensations localized to a specific part of the body. And moving the stimulator around different regions of the somatosensory cortex causes that sensation to move. So if I went stimulated my shoulder part of my brain and then moved on to my fingers, I would then feel some kind of sensation in my fingers. And this procedure was performed actually on an awake patient. But this is possible because the brain itself doesn't have these mechanoreceptors that we discussed, or any pain receptors. So a patient can't feel anything at all in their brain when they've got these little electrodes. So you're able to do this. So then you can just ask the patient, you put stimulation, where can you feel a touch sensation on your skin? Okay, so lastly, I wanted to discuss neuronal plasticity. So this refers to how the brain is able to change. So it was traditionally thought that the adult brain was not plastic at all. but it's now known that it does have a very high capacity to change. And one example of this is how the brain changes after some kind of injury and also overuse. So if you were to perform a task that uses one part of your body load, so if you're playing football and you're using your foot a lot, or if you're kind of playing a video game and you're using your thumbs a lot, the brain will actually change to be able to perform that task better. And so if a body part is lost or overused, the brain can reallocate space, and this is called remapping. So we'll go into this in a little bit more detail. So a guy called Mersnich in the 1980s, he carried out a series of experiments investigating this, and he performed them on monkeys. So he investigated the regions of the somatosensory cortex that are active when different digits are stimulated. And he mapped these different regions using microelectrodes. So before, I said that we had these electrodes in the brain and we were stimulating them. But in this one, it's kind of the opposite. So instead of putting current in and seeing where you get a sensation in the skin, you're touching the skin and seeing which regions of the brain are active when that happens. So it's just the reverse thing that's happening. So one of his experiments that he did was he removed one of the digits. So he took off one of the fingers of one of the monkeys. So he took off the third digit, it was surgically removed. And after 2 months, he redid this in the cortex. So he put the electrodes back in, touched different parts of the skin, and saw which parts of the brain were active when he did that. So if we just look down here at the image, before he did this removing of the finger, you can see there are these different areas of the brain that are activated when you touch finger 1, finger 2, finger 3, 4, 5. And then he removed the digit and did this again. So areas of the cortex that were devoted to the amputated digit, so digit 3, responded to stimulation of the adjacent digits, demonstrating that if a region of the brain is no longer used, the surrounding areas will take over. And this is called cortical expansion or remapping. So as you can see, digits 2 and 4, the region of the brain that's representing that information is got bigger. So this kind of makes sense. If you've got a part of the brain that is no longer used, that's it's a waste of space, really. So you want to be able to change what that part of the brain is responsible for, because there's no longer information coming from that third finger. So other regions take over and it becomes a useful bit of brain again. So then he performed another experiment that was the reverse. So he trained the monkeys on a task where they used 2 of their fingers a lot more than the other ones. So monkeys were trained to perform a task, and after several weeks, the representation of the stimulated digit had expanded compared to adjacent unstimulated ones. So instead of cortical expansion, this is called functional expansion based on a repetitive behavioral task. So as you can see down here, the monkey used digits 2 and 3 much, much more. And so the brain space that's represented for them digits is now much bigger. So it's just the opposite that's happened when you remove a digit. So another VVox now, true or false? Losing a finger can increase sensitivity in the remaining fingers. Very nice, so we've got 100% true so far. Yeah, so that is absolutely correct. If you were to lose a finger like digit 3, you've now got more brain space for digits 2 and 4. So then you actually have a greater sensitivity in them 2 fingers because you've kind of got more power to do so. So lastly, I just wanted to go through something called phantom limb pain. So this is when, these experiments demonstrate that cortical maps are highly dynamic. And this is also shown in this phenomenon called phantom limb pain. So when someone loses a body part, for example, their hand or something, they'll be able to feel these phantom sensations as though their hand was there. And for a long time, it wasn't known what was happening, but it's now thought, this is still a theory, and there's a few different theories, that this is a result of cortical reorganization. So when a limb is lost, neurons in the somatosensory cortex that used to respond to signals from that missing limb begin to respond to other inputs. So stimulation of body areas whose somatotopic representation border the missing limb then lead to phantom sensations. So this was kind of introduced by someone called Ramachandran, and they demonstrated that this remapping by showing that stroking different parts of the face led to perceptions of being touched in a missing limb. So they had someone that had lost their arm, and they found that if they recorded in the brain or looked at the activity in the brain when someone stroked their face, that would lead to these phantom sensations. So this is showing that the area that initially represented the face is now expanded into the region that once represented the hand, because we don't want this to be kind of a dead brain space, because that would just be kind of useless. So stroking the hand leads to this phantom limb pain. So they performed functional MRI, which is a type of imaging technique of the brain, and you can look at how the brain is changing over time. And that revealed that the cortical regions that once reported on this missing limb were now stimulated by stroking the face. So it's a very interesting phenomenon, and it affects a lot of people that have lost limbs. They kind of have this pain. It's now thought that this is a result of how plastic the brain is and regions taking over that part of the brain. Okay, so now just a bit of a summary. The sensory homunculus demonstrates how touch is mapped in the brain. Areas that have higher sensory acuity, like the lips and the hands, have a greater density of receptors and also more brain space to process that information. And neuronal plasticity allows the brain to adapt to changes and also after injury. So yes, thank you very much. I hope you